All right, are we back? Did it start a new video, or is it on the same one?
Let me. It takes a little bit to catch up. One person said it was still buffering.
Yeah, Paradox said it's back, so I think it's
okay.
back.
Well, like I said, if you get it fast enough, it actually
Okay.
keeps the same video. If you don't get it fast enough, then it actually starts a new one. I'm hoping this continued from the old one.
Yeah.
Yeah, this computer's been rebooting lately. I don't know what it is.
No, it's they said it's timed out.
I've refreshed again. Let me take a look.
Maybe I'm gonna have to look at the memory here, and if it can take higher memory, I'm gonna have to get with Druce on it, and maybe I'll see. Again, this is it was a home built unit, so but it's been rebooting on itself lately quite often. I don't know, maybe it's just dust. So what I'm gonna do is maybe tonight just take it outside and just kind of blow it all out, and we'll see.
How many things are on your wish list, Steve?
I kind of took that down. I think I only have one thing on it. I think I have a computer on it, but I mean, I'm not really that. It's not critical, right? I mean, this gets me by. It might have problems, but I don't want somebody buying me a computer. Obviously, Hannah was going to donate one. She had one that she had built for me custom, but she kind of felt my channel was not up to par anymore because I was treating Young Creation as hostile, which I don't think was a fair assessment. But whatever, I love Hannah. But
yeah, we're apparently completely off.
What's that? I'm off?
I said, oh wait, no, wait. Now he says we're back live. What the fuck?
It should be on.
Okay.
I'm streaming.
It should be on. Yeah.
But I mean, if somebody wants to donate an older computer, that'd be great.
Yeah.
I had somebody in Germany offer, but I don't know what happened to that either. But yeah, I'll take a secondhand donation. Because computers aren't really an old, inexpensive one wouldn't be that much. But
hey, you're too late, Steve. I get my old laptop for my nephew.
Oh, I don't want a laptop.
Oh,
I have a little laptop, and actually, it's perfect for just watching TV. I'll watch Couch Tuner. That's all I use it for. I don't need a laptop. I like desk units. But yeah, this thing is probably on its last legs.
I don't know. You'd probably like my laptop. I got the Asus ROG.
Well, I'm so used to a desktop for doing this kind of stuff. I wouldn't know what to do with it.
I got mine's just like a desktop. I got 2 monitors hooked up to it besides my laptop screen.
Yeah, so I guess you can stick in an external keyboard, right? An external LCD.
Oh yeah,
but you got an external keyboard, a keyboard. I got a 55-inch TV hooked up to it.
Oh shit! Nice. HDMI out.
I've got to say, Steve. Anyway, I've got to say that Ronnie's video to you yesterday has got to be some of the stupidest shit I've ever seen come out of his mouth.
Oh, the video that he had.
Oh yeah.
Oh, it. Oh my god, dude! It was so fucking riot because, I don't know if you listened to it just a few minutes ago, Gary, because you came in afterwards, but we had noted that somebody had noted to me, and I remembered it in that video. He made a big deal about me and one of the Evolutionist Bringers, which was how long ago, right? He's going back eons on this shit, right? Where he had
I'm
asked.
pretty pissed. I didn't make the cut in that video.
Did you watch it, Roof?
I did, and I wasn't in it. I was spewing.
Well, this was about dissing on me. I don't know. We'll talk about that in a sec. But he had placed in there a part where he said, he asked me about evolution, and I was just kind of being direct because I was like, look, I know he's not going to understand any of this stuff. So he's like, evolution's a fact. I'm like, yes, evolution is a fact. Evolution and common descent are a fact. Meaning that they exist, meaning that we see common descent, we see evolution. Then he says, something else, and I say, well, evolution is a fact and a theory. And he thinks, because he doesn't understand English, that somehow invalidates my first statement. Because obviously to him, he doesn't realize that it cannot be a fact and a theory at the same time. So if I said it's a fact, if I said no, it's a fact and a theory, I'm somehow changing what I said before, and that's not. It's just clarifying because it is both a fact.
Well, because I don't know whether it's good to really noticeably done stuff, which is like him saying that you Fiona are atheists, which is new to me because I thought Fiona was a Catholic and you're a
agnostic.
Yeah, you're an agnostic.
Well,
I mean,
this-
both of you clarified this like numerous times.
Well,
I mean, I
Gary,
wouldn't
they
mind
have
if it
no-
was the twice, but numerous.
Gary, they have no issue about misrepresenting people's theological positions. They have no qualms about it whatsoever. As a matter of fact, I think that's one of the worst things about the Clown Car Posse is them doing that, because I take people's theological positions pretty seriously. If somebody tells me this is what they believe, fine, we'll talk about it. Like General is a evangelistic theist, right? I don't question him about him being an evangelistic theist. I don't say, you know what, hey, General, you know what, you're actually a Baptist. He'd be like, no, I'm not a Baptist. Well, yeah, you are. Or if Jade's a Catholic, and I say, hey, Jade, you're a Jadist because your name's Jade. She's like, no, I'm not. That's what they do. They misrepresent what people actually do believe. And I don't find that to be something very Christ-like, because it would be no different from me saying, you know what, you're not Christian, Gary. You're Quaker. That's not Christian. And you're like, no, Quakers are Christian. Wouldn't you be like, Steve, you're an idiot if you think that I'm Christian?
Agreed. But also, the thing is that he also misrepresented what two people on his actual side think. Like, he misrepresented what is it Fazel or Fuzz? Sorry, and Hugo Ross said about. Yeah, what they said about the DNA water thing. When he misrepresented that, Hugo Ross said that dissolve and degrade the same thing. That's not what he said.
Because he didn't clue the rest of the clip, right? And I had actually sent that to Fuzz. I had talked to Fuzz quite often, and Fuzz was like, "It never ends, does it?" And I'm like, "No." I'm like, "This is absurd," because we had asked Fuzz that question the very first time, and he said, "Absolutely, they're different." And then the second time when we had asked Ross, he's like, "Why don't you ask the biochemist in the room?" And Fuzz was like, they did. And he explained to them, because that look that he's talked about, the look that they shared. Now, I guess people can read things differently, but of course, I don't think that they know how to read people very well. When I watched that over again, and I watched that exchange, the look that I saw from
Sorry,
Fuzz.
Steve.
What
He said,
do you think,
I don't
Brew?
think they know how to read. Pause. People very well. That was nice.
Well, did you watch that? Wait. When Fuzz looked at him, don't you think it was more the look like, oh, wait a minute, dude. No, you're. Let me step in here and correct this. Yeah, Fuzz realized that what Ross said was not quite correct, but Ross is an astrophysicist. He's not a biochemist. Okay, not the same thing. Go ahead.
Well, also, the thing is that when I had my debate with him, he was using two different dictionary terms. And I've pointed this out to him numerous times. And also, I've pointed out the fact that Fiona knows more about this than anybody in this room, because she's dealt with DNA structure and bacterial and all the rest of it. So probably knows this subject better than anyone. And the fact he's trying to educate her on a subject that she has a PhD in, it's just like ridiculous shit.
Well, I mean, he's infamous for that, though, right? I mean, how many times has he said that, Fiona doesn't know what she's talking about, or Fiona's wrong? The arrogance of these people.
Well, and this is my problem. It's just like, look, when somebody knows something more than I do, I try and tip the hat. I'm a stubborn piece of shit, but at least I try to tip the hat.
Sure, but Gary, okay, we'll go back to Ruhif real quick. Do you guys remember when Ruhif was hanging out with them, trying to explain to them things about the Munchak deer? And he was explaining to Ronnie, hey, look here, here, and here. And Ronnie said, hey, look at all those telomeric repeats.
Oh, dear.
And Ruhif's like, can you show me one? Where are you seeing these things? And he starts like babbling off, oh, look at the TAAG and this, that, and it's just all over the place, right? And I'm pretty sure that was when he started talking about the telomeres for tetrahymena, which I think is TAAG or something. You're not realizing that that's a telomere for a specific species. No, and I don't know if that's where he got. Well, I think he did. I think he had a cursory thing. I think he did read somewhere along the lines that there are different types of telomeric repeats. And so in his mind's eye, he confused them all together and say, hey, look, for humans, it's anything that's with a T, an A, or a G in it, I think.
Well,
I think
actually,
anything.
I am going to be perfectly fair to Ronnie here. Synthase and synthesis do mean two different things. I know because I
right, but okay, in that particular video, I know exactly what I was referring to, and I know for a fact that I think Ronnie, during his discussion with Jake Herder, was conflating ATP synthesis with ATP synthase, and I can make an argument for that, but him just pulling it out of my video and saying, "Oh, Steve doesn't know the difference." Well, of course I do, because that's why I was pointing out the fact that I don't think he did. Right? So I don't know anything about that video that he made the other day about me. Made no sense whatsoever. It was basically him intellectually poning himself, making himself look out to be a complete and utter moron, more so than he usually does. But it was entertaining.
Here's the thing, Steve. I would love you or Fiona or Jade to tell me the stuff I've looked at and tell me if it's wrong or not. Because again, I can only go by what I looked at. And the thing is that unlike Ronnie, I'm willing to take correction.
Well, I'm sure Fiona and Jay would love to do that. I mean,
Because again, what I looked at synthase and synthesis. Synthesis talks about the reaction that certain chemical bonds have with each other, and synthase talks about the initial reaction that talk about the building blocks that led to that particular reaction. I think that's correct.
Synthesis is the process. Synthase is the enzyme.
Yeah, anything that ends in ASE is the name of an enzyme. So ATP synthase is the enzyme which carries out ATP synthesis.
And that's where Ronnie got confused.
Okay, is this where I'm getting confused as well?
I don't.
Yeah, synthase it's the name of the enzyme. Do you not remember in school you did the experiment with starch and how your saliva changes the starch to sugar?
Yep.
Yeah,
because
I did
it's
that.
an enzyme which is salivary amylase. So that's the enzyme. Amylase, salivary amylase.
Right,
So anything like peptidase, transferase,
Collagenase,
they're enzymes.
Ligase,
ligase, DNase.
Yeah.
which I'm having trouble getting, by the way.
No, but excuse me. What I looked up, I mean, this was from my cursory Google search that I did of certain papers, and the meaning I was able to get from them was that synthase meant basically the initial start. So, like, let's say, for example, you had somebody punches somebody in the arm. That would be a synthase action because your body's going to change and then synthesis.
ATP synthase. This means to produce ATP. Excuse me, ATP synthesis. Sorry, ATP synthesis just means to produce.
It's the international nomenclature system for enzymes. That's what it is. That's what synthase, an enzyme which synthesizes something.
Right.
And the clue is usually in the name.
Yeah, I'm just going by what I read, Fiona. I mean, that's the reason that I was asking you yourself, because you know more than I do.
Yeah. No, it's just to do with the name of the enzyme.
Okay. You see, because as I said, the paper I read was, I can't remember what it was. I think it was like some like 2004 paper on about how body reactions to different different structures. So, like, let's say, for example, if you get punched in the face, what happens? And it had what synthase meant in that particular way. Maybe that was what I was reading wrong.
Synthase means to produce. So ATP synthesis. Synthesizing. Synthesis. ATP synthesis means to produce ATP because it synthesizes it, right?
Yeah. But synthase
is the enzyme.
Is the enzyme nomenclature.
So Gary, what do you think ATP synthase would produce?
Enzymes.
ATP. Then the clues in the name.
Again, I'm just going by what I read. Sorry.
Well, I'm just telling you.
This is the thing, Fiona. It's like I'm an English grad reading a science paper. What I fucking know about it is like the end of the earth. But probably what you would get out of it is probably a lot. If it was an English paper, I'd probably get a lot more of it than you would. And that's not an insult about you. It's just again, this is my stupid English.
I'm finding out that I a while back I found I have to see if I can find it again, but I found a really good list of like prefixes and things and words in biological terms of what they kind of mean. Right. And a lot of times you can actually determine what it does by the word itself. For example, like he just mentioned the word ligase, right? Means from ligate, right? And
I'm just going to give you the definition. In biochemistry, a synthase is an enzyme that catalyzes a synthesis process.
And do you
So
know
it's
what that
not a
means, Gary?
verb. There isn't a verb to synthese.
Synthesize.
Yeah, but not to synthese. So that's the definition. Synthase is an enzyme that catalyzes a synthesis process. ATP synthase synthesizes ATP.
And
Yeah.
Gary, a catalyst is just something that makes a reaction go faster.
Yeah.
Basically.
Okay. I mean, as I said, like, you know, this is the problem,
Yeah.
is that when you're looking at a paper and you're not really used to reading it, you're going to misunderstand it, I think. And this is the problem that I have when I try and look at something that I'm not familiar with, especially like with science paper and stuff like that. I'm not used to looking at the research and what it means and stuff like that. I mean, I get it. All I know is that they are actual terms that I actually looked up because I actually want to know whether they're actual terms, you know, because despite what Ronnie thinks, I actually like to look up whether the terms he uses are correct. And I don't.
That's the thing. Ronnie doesn't look it up. Ronnie just assumes he's understood it.
Yeah, I agree with that. I mean, the thing is that I don't get why he badmouths you. You see, because what I try and do is I ask you not because I'm questioning you, but because I want to know.
everybody.
Because I know that when it comes to science, you've done it for numerous years in a laboratory, and you've done the research, blah blah blah. And because I want to know from somebody who's educated, see, because then I get to fill in the gaps in my knowledge. I don't think it's a bad thing on me, but if I don't know
But
something.
Ronnie doesn't think he's got any gaps, Gary.
Right.
That's the problem.
Yeah, he really doesn't. I mean, he projects it on anybody else, right? He thinks Fiona doesn't know anything about this. He thinks that even myself, who is a layperson, he thinks I literally know nothing about this topic. And I'm like, dude, this is something we learn in college. This is not that complicated stuff. Anybody who has any kind of biology in college has to learn, you know, what the electron transport chain is and things like that. He thinks it's PhD level. He thinks that if you know anything about it, you're intellectual elite. No, it's the foundation. Matter of fact, most people that are probably, you know, heavy into biology like Fiona probably wouldn't even remember something along the actual mechanism, that kind of stuff, because it's so basic. It's just, you know, you use it and forget it. Like Fiona, could you remember all the steps in the Krebs cycle, like off the top of your head?
Good a heck is like. It
Why?
starts at pyruvate. Oh, can I remember? that?
Pyruvate.
Off my head.
There's pyruvate in there. I remember that.
Yeah, I mean, this is the thing. There's no need to memorize stuff like that. And having it memorized means absolutely
Bingo.
nothing.
That's why I thought it was funny when he said
You know
memorize
what
a
I mean?
periodic
Unless
chart.
you're using it every day, there's no point in having it taking up space in your brain.
What an expert would be like Fiona is, though, she can look at it now, but you know, she doesn't have to memorize, but she can look at it and go, yeah, of course I understand how that works. And that's what an expert does, right? You don't have to have these things memorized. You just have to know how to explain them when you do see them.
Well, and you need to know how to use them.
Mhm.
A while back, you know, I was following some of Red's stuff, and somebody tried to complain about how a rocket worked. You know, it's impossible.
Well,
You know, rocket thrust. So I decided to look it up. Okay, looking it up online, and I'm seeing this equation, and I'm going, okay, I don't quite understand that equation. Where did it come from? So I looked up that equation to find out where it came from. I actually have a whole couple of pages here where I went step by step backwards. I want to go forward with it later because I didn't understand where those certain things came from. But I understood, you know, what the equation looked like. So I went to look at the parts of the equation, and I found other equation, and I was like, okay, that's really interesting. I understand it, but I don't know where they got this part came from. So I went backward again. So I went back through the list, go back to really, really basic physics. Once I got there, I was like, oh, up here works. You generate this right here, and then you take that and you generate A, okay, and then you put A in this other equation and you get B. And then you take B and you put it in this other equation and you get C. You take C and you put it in this other equation, and that's how this thing works. Oh, it's really cool. The thing is, I remember a lot of my physics, but I was able to work my way backward. And as I did, you know, all those little brain cells that I haven't used for physics for a long time started to spark to life. Because I understood the basics, I was able to figure it out. That's the same thing with like chemistry, with a lot of the stuff that's come up. I can go back and somebody can, say something and I'll go, okay, I'm not sure how that works, but I can look it up. And if I have to go back, even back to, how do you make hybrid orbitals, which I am actually pretty good at, but, I can go back and understand the entire process because I have that education. I don't have everything at the tip of my brain. You can't just pop everything out. But having studied it from the ground up allows me to understand it, allows me to be able to dig into it and figure it out.
Right.
And that's really what this is all about.
Yeah, give me 30 minutes, maybe 30 to 45 minutes, and I could probably explain to you SP1, SP2, SP3 suborbital hybridization as well. Because, I mean, I learned it pretty decently, but of course, it's not on the tip of my head of starting off, with which arrow goes where, okay, and, moving, what needs to be the change in this orbital to produce the hybridization. But I could theoretically do it, but I only need about a half hour to really review it and go, yeah, of course, I remember how to do this because we've had this before.
Well, and even drawing Lewis structures. I'm sure there are.
I can't. Well, Lewis dot structures.
Yeah.
You had Vespers. You had Vespers theory. Okay.
Yes, and I'm sure there are a lot of people. If you were to say, how to draw a Lewis structure, would look at you blankly, but the minute you drew one, would go, "Oh, I remember those."
I don't think they would. I mean, again, I would have
to. If you took chemistry and you drew a dot structure, you said, "Well, do you know what a Lewis structure is?" And they go,
"Sure." I guess if you took chemistry,
and then you draw it. That's what I'm saying. If someone took chemistry years ago, and you say, "Well, you know what your Lewis structures are," and they'd go, "No."
Right. But
and then if you were to draw,
but do you think you would remember where some of the dots go on some of the things? I mean.
Right. Well, no. What I'm saying is, I'm not even talking about that. I'm just talking about the name Lewis structures.
Oh yeah. Of course.
People would not remember the name, but the minute you drew a Lewis structure, they go, "Oh, I remember those dots."
Yeah, probably.
And then it would probably just take, depending on how much attention they pay in chemistry class, it probably wouldn't take very long for you to say, "Okay, here's oxygen," right? Okay, you remember what oxygen looked like?
Yeah.
Okay, you probably don't.
Two hydrogen with it, and then two dots because there are two unpaired electrons. One unpaired.
Right.
Yeah,
yeah, and well, because you have the one unpaired electron. But the point is, most people would be able, going through it, they go, "Oh yeah, I remember." And as I said, it's like there's a little part, you have this stuff in the back of your brain that you just don't use, but it's sitting there. It's in a closet. It's a little dusty, maybe has some cobwebs. But once you open the closet door and you vacuum out the dust and you chase the spiders away, oil it up like the old creaky Tin Man in Wizard of Oz, and then it gets working again.
Yep. Did we lose her? Or...
Yeah. No, still there.
Okay. Yeah, no, I couldn't agree with you more. I mean, I think a lot of us probably do have a lot of stuff we've long forgotten, but if we could just, have a little bit of review, it would be, of course, I kind of remember that. But I think they put too much emphasis on rote memorization, like it's that meaningful. I think conceptually to learn something is far more important than just having something memorized, right?
Mm-hmm. And if you remember that one hangout where I was in the live chat, I think everybody who was there remembers it. Where Ronnie was saying about how he has the periodic table memorized, but he couldn't give me the first three orbitals.
See, and I remember doing that in like 11th grade. I'm serious. Yeah. It's like 11th grade we were learning about orbital structures.
Right. And he couldn't tell me how the periodic table was arranged and how we could derive information from it. It's a masterful thing. And I was even giving him hints saying, okay, what do the rows mean? What do the columns mean? He couldn't answer those simple questions. Having the I don't have the Christ. I don't remember where a lot of stuff is. But I can look at it and I can tell you, if someone were to say point to an element on the table, and even if it was an element I was unfamiliar with it, just because of what's column and row it's in, stuff about it. I could make good guesses about it.
Yeah, I mean, look at the things even like sodium and potassium and those things that react with water. Is the same column. Look at the noble gases, same column. I mean, you can make some kind of inference by where these things are in the periodic chart.
Yes, exactly, and you can have a general.
Sodium,
you have a good idea of how they're going to react and what they're going to react with, and what what electrons are, are they going to, you can point to something electronic on a table with the exclusion of the stuff at the bottom, and is it going to make an ionic bond or a covalent bond?
Well, do you remember when Ronnie said something about that? When he said that all ionic bonds will break down into sodium and chlorine.
Yeah, that was just hysterical.
Now, again, I'd like to know what he meant by that. Do you take the hogtie approach where he probably was just misspoke, or do you actually think that he thinks in any ionic bond it'll break down to sodium and chlorine? Because that's what Drag was trying to say. Because Drag was trying to say he only thinks there's one kind of salt, table salt, basically.
Well,
don't know.
Here's the thing. Like, Hogtie has numerously said that I get the theory of evolution wrong. When no, I don't. I express how how it's expressed in numerous books I've read. For example, I know it from it explains the change of allele frequency in a population over time. Basically.
Hey, Lemur has a question for you, Fiona.
Sorry?
Schrodinger's Lemur has a question he would like to ask.
Schrodinger's lemur.
Is it alive or dead?
No, is Schrodinger's lemur in a tree or not?
Oh, there you
He
go.
says he wants an invite. He's got a question.
He's
for
already
you.
in here.
Just don't look up the tree, and I'll be fine.
Yeah.
Fiona, I was thinking about my understanding of what a species is the other day, and I found it was tied to the idea that basically if two things are different species, then they don't have much success making offspring. And then I thought, well, wait a second, and so you get speciation events where things get separated, and over time they diverge sufficiently that they can no longer create offspring, and so you look at those and go, okay, they're now different species. Mm-hmm. But then I started thinking, well, wait a second, there are asexual things in the world, and so my definition of species just has no applicability to that at all, because they never have to get together to create offspring. What's a species for an asexual entity?
I mean, I can only talk about bacteria, and I'm not going to give you a hard and fast dividing line, but they work it out. I mean, at one point they were able to group like bacteria based on physical characteristics, biochemical reactions, and dividing line, confirm it by working out. They work out GC content, okay, and that was the proportion of the genome that contained G's and C's, okay. Because you find that related bacteria would be very similar. But now they can do that was in the days before they could sequence them. So they now do it based on the DNA sequence. So you have all of these bacteria that have been classified using the old method. And it's then when they look at the DNA, most of those relationships are panning out. But it is, I mean, even with whether something breeds or not, again, I mean, it is a bit of an arbitrary line that we've decided, because we've decided what we mean by species. So look up, go Google Berge's Manual of Determinative Bacteriology, and see if you can find any information on the much older versions of that book.
Okay, thanks. That was a good answer. Okay, so we're basically looking for differences in the genome which are sufficient that we think that, I don't know, we think that if they were, for instance, actually a sexual species rather than an asexual species, then we'd probably see sufficient difference that we would call them different species. Is that the gist?
Mm-hmm.
I put it in live.
Basically, yeah. I mean, yeah, because you get, as I say, we designed this classification system, and the organisms haven't read the books.
Sure, Feynman said it best. I think he said we can call nature whatever we want. She doesn't care.
Exactly.
I
But
find
that's
that-
we have to accept that, and we go with our working definitions. Now, if we needed to change them because they didn't work, then that's what we would do. We would change those definitions to actually suit, because there is no point in using a word when it doesn't accurately describe what you're trying to describe using it. And I think a lot of people get confused because they think that in some way, life itself has categorized itself into all these lovely wee pigeonholes for us. Well, it has sort of up to a point. But of course, there are always going to be exceptions to the rule as well.
This is interesting, actually. I'm looking at this Burgess manual, and it's like how bacteria are named and identified, and it gives all the Latin prefixes. And I'm definitely bookmarking this. I put it in the live feed. And have you read this, Jade?
That's the Bible for bacteriologists.
This is interesting.
Right.
It's a fantastic. I mean, it's not the type of book you sit down and read.
No,
Right.
no, it's
It's
not,
got a
honestly.
table, diagnostic tests, descriptions, of the actual characteristics, like gram-negative diplococcus and things like that.
Yes.
But it really helps you to understand why they make the decisions that something asexual like a bacterium belongs in a different species or a different genus.
So would this book help you? Like, I mean, when you look at a word, right? Let's say you've looked at a species you've never even heard of before, because you can kind of determine a little bit about it because of the Latin words that are being used to describe that particular thing.
No, I think what happens is it's when you look at the genus name, then you know where it sort of fits.
Yeah, that's what it's on page like
You know-
19. It's talking about the different adjectives used for genus to describe whether it's masculine, feminine, neuter.
Or that? No, you don't need to bother with that. I mean, that's very old-fashioned, but it does describe how and why they've ended up named the way they are. There's various naming protocols.
And now you get to name things anything you want. You can name something after Trump.
Yeah, Arwinia caratovera, subspecies Atroseptica, has now been renamed. It's been put into a different genus, Pectobacterium.
Yep. Well, there used to be something, I can't even remember what it was, as a type of clam that was called abracadabra.
Yeah.
Abracadabra.
But then it discovered, it really was. But then they discovered it really belonged to a different genus, so that's no longer. What I wanted to say though is, the determinations for species in bacteria, in prokaryotes, is different than that in eukaryotes, because they are. But even in eukaryotes, in budding, budding isn't perfect. That's just one type of asexual reproduction is budding. And the best example I can give is in plants, because it's easier to understand. When plants produce buds, anybody here has ever, remember way back to when you took biology, you probably looked at a onion root slice or something. And the reason they did that is because the replication of cells, the division of cells in things like an onion root or in any growing tip, and an onion root is a growing tip, is really, really rapid. So they take these onion root slices so that you can see all the different mine just went blank. Mitosis. You can look at all the different stages of mitosis, right? Because the onion root is growing so fast that at one time, in one slice, you can see all these different stages of mitosis. Well, the same thing happens to regular buds, which is how we end up with things like apple, or it's how we ended up with the navel orange, because a bud in rapidly dividing has an error in it, just like in it's a genetic error, so you end up with an entirely new variety. And eventually, if that one produces, because again, in oranges, we have the sweet orange, which itself is an interesting, came from a hybridization and a polyploidy, and it budded, and we had this bud that created a navel orange. The navel orange itself, then in budding, created the Cara navel orange, which is a pinkish navel orange. So here we have a variety 2 steps away, caused by budding. But the same thing happens in certain animals, like if you're looking at hydra or a sea anemone. But sea anemones duplicate by both budding and bisexual and asexual. But when they bud, again, you have a rapidly growing portion of animal, whatever it is, a rapidly growing portion of animal plant. And whenever you have rapid growth, rapid duplication like that, you're going to have some genetic errors, just like you would in in sexual reproduction. And so you can get a different variety. And if that variety then produces a different variety, and if that one, at some point, you're going to be so far from the parent that you're going to call it a species. It steps in between. So that's a kind of how speciation can occur in reproduction in eukaryotes.
Yeah, I understand that. Sorry, I'm getting looped back. I'm getting an echo.
Oh,
That's better. So I understand that it'll all happen slowly, and that the definitions we place are our own. They don't have to be honoured by nature or anything like that. I guess you could almost say that a lot of science is a pursuit of improving our definitions. I guess that the problem with plants is that you really do need,
excuse me.
in order to call two things different species, you almost have to have some of the things in between almost have to have failed for some reason. Otherwise, the spectrum is utterly full.
Well, in plants, the thing with plants is a lot of plants have come about by hybridization and polyploidy, and sometimes both. Polyploidy is when you have the entire genome being duplicated. For example, modern breadwheats are the example of two different, and they're very different species from their ancestor. Originally you had a wheat that was something like einkorn, bit of wheat, and it only has 2 sets of chromosomes. It crossed with a grass, an Agalops grass, and from that we got a wheat called emmer. It has 2 sets of chromosomes, and then emmer crossed with another Agalops grass, and it resulted in modern bread wheats, which are hexaploid. They have 6 chromosomes. So it's a completely different species because of these different steps of hybridization and polyploidy. Speciation, it is estimated that from 30 to 80% of plants are polyploid. And the reason that you have that wide gap is because we haven't done genetic testing on a whole lot of plants. So their estimation is a minimum of 30% and a maximum of 80%, given what we see from plants that we're doing now. So plants just, Luther Burbank even marvelous experiments, and some of which have been lost, unfortunately, in crossing plants that are distantly related. And of course the crosses don't always work, but occasionally things just line up so that they work. And so this is this is how a lot of species have come about in plants by hybridization and polyploidy. Also, because they've not long ago did the genome for goldfish and discovered they're polyploid. So somewhere there was a genome duplication in the ancestry of goldfish, and that's how we get goldfish. So there's a couple things. First of all, I think a lot of the younger creationists are just really too hung up on species and speciation, because it can be fuzzy. What do we mean by species? I just like the basic term isolated breeding population. Inherent in the breeding is that it produces fertile offspring. But what does isolated mean? If you go to anyway, if you look at the birds of paradise, okay, the birds of paradise are could theoretically interbreed. Most of them, they don't. The reason they don't, and they're all very, very colorful, but it's less the fact that they're colorful than it is they have a specific mating ritual. The mating rituals of each different species of bird of paradise are so distinctive that if a male bird of paradise from one species were to do his little ritual to a female from the other species, she'd just look at him like, what the fuck? And there would be no mating. It's not that maybe somebody ate the wrong fermented berries and didn't mate or something. I mean, it's possible it could happen. But generally speaking, they are a separate species, even though theoretically, physically interbreed and produce fertile offspring. They don't, because their isolation is mating ritual. So they all live in the same area. So for the isolation can be geographic, it can be behavior, it can be coloration, it can be a lot of different things.
And you'd have to include, if that was your definition, you'd have to include incompatible genes as being a form of isolation as well.
Yes, you would. Yeah, it's just not going to work. It's like the the salamanders here in California. There's a genetic incompatibility there. The ring species, and I always mispronounce it, so I'm not pronouncing it. Of salamanders here in California, there's a it's not going to work. And another problem with younger creationists is because they will go, yes, but there was this one case where they crossed. Yes, we know that. We know that even though you may have a basic genetic incompatibility between populations, that's the part they don't get. You have a genetic incompatibility between populations. That doesn't mean originally going to get compatibility. We know that horses and donkeys are incompatible. They're two different species. They produce sterile offspring. Guess what? Every once in a while, rarely, but it happens, you get a fertile mule.
Yeah, it's like 1% or something. It's really low.
Right. So just because it can happen doesn't invalidate the fact they're two different species, because we're not talking about individuals, we're talking about populations. The population of horses, the population of donkeys, they can't interbreed and produce fertile offspring. But that's the thing, they're so hung up on species and speciation and not understanding that, yeah, scientists know that it's a little bit amorphous. And when you get to subspecies, it's even more amorphous, and it can change as we go along.
It's a little arbitrary, right? It's a little arbitrary. And there's dozens of different things that go into what qualifies as a new species, correct?
Right. And it's our definitions, and that's the other part they don't seem to get, which is why abracadabra no longer exists, because it discovered that it doesn't belong to the genus Abra. That happens. It's just we do more studies and we go, oh, wow, gee, look.
Yeah, they've done that with spiders too. They found some spiders that they're like, oh, you know what, this is not in the right, genus.
We don't mention spiders because.
Yeah, Fiona hates spiders, doesn't she?
Yeah, I know.
But
yeah,
I hate them too.
Yeah, I love them.
Oh, you're crazy.
Sorry.
I just love that the magic clams vanished in a puff of logic. That was great.
Yeah.
By the way,
the clams still exist. They're just no longer abracadabra. I know, that's great, isn't it? It's kind of a bummer because there's abracadabra was such a great name. It's too bad that yeah, you're right. It disappeared in a puff of logic. That's great.
I got to make an announcement.
Have you looked at the names of some of the mutations of Drosophila?
Madagascar?
They're fantastic.
That's the only one I remember.
There's one of them that gets very badly affected by alcohol. It's called that mutation is called a cheap date.
A cheap date?
Yeah.
The cheap date mutation. The cheap date mutation.
Yeah, cheap date mutation. Go and look it up. Some of the names. But you see, that's where they got Sonic Hedgehog from.
Yeah.
Drosophila mutations. That's why there's a Sonic gene. The mutation was called Sonic Hedgehog.
Yeah, cheap date is the name of the mutation found in Drosophila melanogaster.
Drosophila.
Drosophila.
I can't think, but yeah. But look up. There's you'll find it online that there is a list of all of the names of the mutations in Drosophila,
That's
and
funny.
some of them are hilarious.
14 of the funniest fruit fly gene names. Here, I'll post
Well,
a few
and not
of
only
these.
that, you can look up.
Read them out.
You can look up, like funny or interesting scientific names because scientists have a sense of humor.
Oh,
And you look up some of these names of species, and they're hysterical.
The Cannon Barbie
And
gene, the Indie gene,
Yeah,
Boss gene, Cheap Date, Lush, Halloween genes, 18 Wheeler, Tin Man, Clown, Van Gogh. I didn't know there was a Van Gogh gene.
is it
Dachshund,
missing in here?
Dreadlocks, Swiss cheese mutation, the hedgehog mutation. Yeah, you're right. Yeah.
Yeah.
Anyway, I want to make a quick announcement.
they're brilliant.
I'm having a
Brilliant.
hangout in 12 days. It's going to be my birthday hangout. It's on Valentine's Day. If you do not have a significant other and you're bored, then come hang out with me on Valentine's Day. If you do have a significant other, do not join me. Go play with them for a while, or at least get done and then come play with me. So yeah, I just want to make an announcement that I will be having a hangout on Valentine's Day because it's my birthday, not because it's Valentine's Day.
I'll be there if I can make it.
But have sex first with your significant others, then come, and
Can you count a hooker as a significant other?
sure. Oh, absolutely. Yeah, that's your significant other for half hour to hour, depending on your staying power. But
So to be clear, we shouldn't be logged in with the mic on whilst we're doing that.
when we're off air, I don't give a shit. Everybody comes in, they're like,
So
Okay.
I've got a question about this polyploidy thing. I recently learned that it's possible to correct various problems in human offspring by giving them more than one set of mitochondrial DNA. Is that would you be a polyploid if you were born to the result of that?
Well, if it's just okay, polyploidy is just a variety of gene duplication. Okay, gene duplication is when you have more than one of the same gene. Polyploidy means you just have all of them or most of them. I don't know how you would call that because you're doing it artificial, or you're just adding. I think polyploidy really only refers to nuclear DNA, not
Usually does.
DNA.
yeah. Wasn't there? There are humans with whole genome duplication, right? But they don't usually survive. They're not viable, right?
As far as there are people who might have a gene that's duplicated and it doesn't do anything. Remember,
But not whole genome duplication.
I thought that I've read somewhere that there have been instances of humans, babies with whole genome duplication, but none of them ever survived. But
Are you talking about sort of XXY and
the whole
and so
genome,
forth?
the autosomatic,
Whole
everything,
genome.
the whole genome.
Whole genome. So that instead of 46 chromosomes, they would have double that. I'm going to look that up now.
yeah,
I
don't
don't know
know.
where I've read that from, but maybe somebody can look into that.
Single gene duplication doesn't necessarily mean anything because one of the genes can be turned off. It's like in women, we're double X. And I think
That's
Fiona could probably explain it
Is that
better than I.
Klinefelter's disease?
We're normally double X. All women are double X.
You talking
Yeah, no,
about
triple X,
X
yeah.
inactivation?
Right. But we have two X chromosomes, but only one of them operates.
Not that simple.
Well, I mean, okay, yeah, I was going to say good. Somebody can explain it better.
This thing. So there's.
You're breaking up.
I think it's the. Sorry.
I think you're breaking up.
Hold on. Let's see if I can fix my mic.
Yeah, it sounds like a loose mic connection, loose mic cord.
Yeah, Klinefelter's
Right,
is
I've got
XXY.
some information
I
for
remember
you.
that. Did you okay? Did you find out what I was referring to?
Okay, true polyploidy rarely occurs in humans, although polyploid cells are in highly differentiated tissue, such as liver parenchyma and heart muscle, and in bone marrow. Aneuploidy is more common. Polyploidy occurs in humans in the form of triploidy or triploidy with 69 chromosomes.
And then triple X for the sex chromosome.
Yeah.
Okay.
So 69 XXX and tetraploidy with
this-
92.
Is this any
Sometimes
better?
called 92
Yes.
XXXX.
Yeah.
So
Yeah.
anyway, with the X chromosome,
Yes.
so since males only have one, the genes need to be able to do with one copy everything they've got to do. But then if you've got two copies in females, that's suddenly too high of a dose, which is why you need to inactivate one of them. And this happens, I think it's at either the 16 or 32 cell stage, and it's sort of random cell by cell. And so females end up being chimeric. They have a mosaic pattern of X inactivation, calico cats, for instance.
Right.
And so it's not as easy as saying that one gets inactivated
Yeah,
because it's not the same one in every cell.
Right. We're all
Yeah.
chimeras, or which
Mhm.
I think is fascinating because you could point to a part of the body, and depending on what tissue that arose from, the one X could be activated in your liver, another X could be activated in your skin, and different X could be activated in your heart. It's just kind of interesting.
Well, was actually so like on the skin, it's actually a patch of skin will have one X activated, and then another patch may have a different X activated.
Yeah. Yeah, I
Yeah.
didn't remember at what level it occurred, if it was like complete organ or you're right about the skin. I forgot about like you said, calico cats, which is why they chose a calico cat for cloning.
Mhm.
And that turned out to be more interesting than they thought when Cece was born. Because Cece isn't calico.
Hey,
I
Christopher.
love the name they gave that cat, CC,
I
because
know.
it stands for copycat.
Copy,
I know.
copycat.
This is brilliant. Yeah.
And they even let Cece have a litter of kittens just because just to check her out, and everything worked properly, and then they had her fixed.
Hey, Christopher, why I got you in here?
Yeah.
Just because it is the topic of the hangout. Did you ever see in that entire thread at any time C. Brown answer my question about where did Mary Schweitzer say she found red blood cells? Because you've been in that thread since the beginning. So I want you on record.
To my
Did you-
knowledge,
did you see anything on it?
to my knowledge, he has neither admitted that she never said that, nor has he provided any evidence that she did say that. So, I guess there you go.
Yep. Well, the bit of evidence that he produced that said that he claimed proved she said it was that they looked similar to or seemed to look like. In other words, she hadn't said they were.
Right.
I mean, my son looks like me. Doesn't mean he's me.
And so, honestly, I mean, I kind of feel like we're sort of getting caught up on the wrong point here because, I mean, biological remnants that there's evidence have been preserved, right? So we have evidence of preservation of some biological remnants. I think that's pretty clear. Most people can agree to that, right? Is that anybody take issue with that?
Nope.
So the thing with his argument is the point he's trying to make is that he thinks that you cannot have long-term preservation of biological compounds.
But he doesn't give a reason as to
Well,
why.
that's not. But he's
Right.
also.
And so that's the unsupported premise that he's got. And so
But no,
if
he's
you-
also.
sorry, go ahead.
The problem is he's also saying that she said these are intact red blood cells.
Yeah, I think that is important.
Well,
and
well,
she's
yeah, but
never
I mean,
said
he's got-
that.
these guys don't really have the comprehension skills for language that we might expect of others. So, I mean,
That
I
doesn't let
think
him off
that
the hook,
if we,
though.
No.
I mean,
And he's
no,
also he's giving us
it
quotes
doesn't.
about
But
blood vessels, not blood cells.
Yeah,
He
that
doesn't
was
know the
funny.
difference between the two.
Yeah, that was
yeah,
funny. Come
so
on,
this is
Christopher,
what I
admit
mean
that.
about comprehension, because I think that he doesn't get the difference at some level.
Uh, okay, to
But,
explain to people
but
what
this
happened was
is going
we
to
asked
the
him for
right
we asked him for evidence that she had said that in the papers reporting about red blood cells, and he talked about blood vessels, and I'm like, well, nobody's questioning that that there's blood vessels there. That's what they were analyzing. That's what she did analysis with ostriches, ostriches blood vessels, but
So,
that's
I mean, you
not
can
the
understand
claim.
if he doesn't see the difference between the two, you can understand
He
why he
doesn't.
would have done it,
Dude, a
right?
second
So,
grader understands the difference between a blood vessel and a blood cell, I would think.
well, I'm not sure how far his education in biology actually went.
I don't think he does see the difference.
So my point is, I mean, yeah, he's made a false claim regarding what she said about what. But the thing is, even if we could conclusively demonstrate, 10 years from now, we can conclusively demonstrate that, yeah, okay, those are red blood cells that we see there. So what?
Well, that's the claim I've said
Exactly.
too. Well, the statement I've said. I
Right.
said, look,
So-
if that was red blood cells, why would it make a difference? I don't. You're right.
Yeah,
I mean, that is a
only-
bigger argument to be had.
The
Explaining
only
why it
reason
matters.
it matters, yeah, the only reason it matters is if you already accept his usually unstated premise that such things cannot survive that long, which he has not supported in any way at any point.
Correct.
So,
I think though we have to sort out the communication problem first.
I'm not sure
Because-
you can.
Well, exactly. If he doesn't understand that saying that something seems to look like isn't saying this is actually, then there's a real problem there because he's not reading the words that are written down. And I've come across that quite a few times with him. He doesn't read what is written, he reads what he wants to be written.
Can I ask about what level the he thinks the impossibility occurs on? I mean, I expect molecules, even quite complicated molecules, to last a long, long time unless they're in the presence of something that'll oxidize them. So it can't be that he thinks that the molecules couldn't last that long. I kind of expect cells to keep their shape over a long period of time, but maybe to break down under a normal environmental pressures. But these things did keep their shape, didn't they? So what exactly is the level where he thinks it's impossible?
Well, that's at least somewhat difficult to pin down. But he's posted a link to a basically to a paper that's talking about a different paper that talks about the fact that somebody did experiments with collagen and found that they could expect collagen to hit the limits of detection after about 3 million years. The issue is that was a gelatinization experiment. And if I understood correctly, firstly, this is unpublished data from this group. But if I understood correctly, this is purified collagen. So none of the normal minerals from bone were present when they did that, much less the iron from heme or whatever else. So he was saying that you couldn't have collagen past 3 million years.
But he's not provided any evidence for that.
Well, he provided a link to this paper that cited unpublished data from a group that does a lot of research into collagen. So I trust that this group actually did the experiment that found that after 3 million years, purified collagen would be fully gelatinized to the limits of detection. But that actually doesn't support what he's trying to say, because he's trying to say is that collagen in situ would no longer be would be beyond the limits of detection, having been degraded so badly after 3 million years. And that's not what that paper says at all.
But one of the things that has really annoyed me about that thread is when he started talking about Lil Miss Mary going to her supervisor. Lil Miss Mary. That really annoyed me.
He's arrogant as fuck.
Yeah.
I mean, just super arrogant. And just
I thought that
condescending.
was, you know, you want to have a decent conversation about science. You don't do that.
Well, that's why I wouldn't allow any of them in there, you know, while she was in here, obviously.
No, I mean,
it would be
you
very
just
disrespectful
can't.
to her. Yeah, I can't
But
take
the
the chance
other
of them
thing
being
that happened in
disrespectful.
that thread, which I think goes to this problem of communication, is that's where he also started getting on about Neffy wanting to debate me, right? And
No, I'm sorry, he was going on about Neffy wanting to debate me. Not to be arrogant, but that is what he said.
I know, further up the thread, he first brought it up.
Yeah.
Way back.
Yeah, he did. He
He
wanted to
brought
debate
it
Fiona
way,
first.
way back. And I issued my, said, I've had a challenge outstanding for quite some time.
Oh, I'm sorry. Yeah, you're right. And then he forgot about that.
Right.
And then
yes.
Okay, right. I'm with you.
And then he, after saying Neffy wasn't interested in debating me, he started again about it. And I'd already given him the links to the full paper that I said I would debate Neffy on. And everybody knows that the paper is called Disease Risks Associated with the Translocation of Wildlife. Yesterday, C. Brown came back posting a link saying, yeah, well, this says there's a disease risk associated with it as well. He's made the same mistake as Neffy.
He linked the same paper?
Different paper. But he was saying, but this confirms that there's a disease risk associated with the translocation of wildlife.
Okay, so nobody questioned there was a disease risk associated with the translocation of wildlife. Nobody questions that. But that wasn't.
Yeah,
that's fucking hilarious. Oh my god!
And the thing is, way back when this first started, I gave him the paper, the name of the paper. I gave him the link to the full paper, and I gave him the link to the video on my channel about it. And he came back to me saying, so you want to talk about chromosomal translocation? I was like, no, that's not what the paper's about. He's made the same mistake as Neffy.
That's fucking hilarious. But Fiona, this paper says right here there's disease risk associated with translocation of wildlife. Well, no shit.
Yeah, I had to point out to him the definition of translocation for wildlife was given in the paper.
In the abstract,
but it involved darts, vets, ropes, lorries.
Yeah, it actually.
And there's nothing seriously bad.
Oh, that's funny as shit. Oh my god, I'm rolling.
But that's what I'm saying. If you go to the trouble of providing the actual paper, it gives the definition in the very first line of the abstract.
Yeah, so something like the
still
definition.
Doesn't get it.
Yeah, so something like the lines that the definition is like moving of a indigenous species from one region of their habitat to a region outside their. I mean, yeah. I mean, I read that abstract of the paper, but it's so funny that he's so potato that he's not even understanding what the argument really is. And this is the funniest thing about the young Earth creationist people, that I should say the Clown Car posse in specific, that even when they have a gotcha moment, they think they have a gotcha moment, and that gotcha moment is basically them owning themselves.
Yes.
Even though they think that oh look, I'm proving Fiona wrong, and then you look at what they have and like, you're fucking stupid. You're just stupid.
So I mean, so CRISPR, I do understand, you know what you're saying there. That, I mean, he does need to show that this claim of his, it can't last that long. Where can he show this? But I think the bigger problem is that he doesn't comprehend what he's reading. And this is why we keep getting provided with, to supposedly support his position, that is about something else altogether.
Yeah, and I think that might be just a point of disagreement between the two of us, because I think that I'm willing to move past that ad arguendo to just talk about, well, wait, can these things survive that long? Let's just talk about that. Because I think I can get to some science there.
And that's fair enough if you want to take
rather
that
than-
route. Yeah, I
Yeah,
mean,
because I was like bickering back and forth about whether or not somebody said something.
well, I think
when
that's
clearly
important.
they didn't say it. Well, I mean, like clearly they didn't say it. You think they did? You're wrong. Let's move on. I mean, that's
but he won't admit that he's wrong. That's the funniest part.
well.
Yeah, I agree.
Yeah,
Look,
they never admit that
I think the
they're wrong.
science would be more interesting for him to explain his science of why he thinks these structural proteins and other things will break down under all conditions, right? And I think you had noted to him, CRISPR, that we don't know what all conditions would even be. Wasn't that what you?
Well, yes. So the point I tried to make to him was that the first step in science is you make an observation of something that needs to be explained, right? Like if you don't have anything that needs to be explained, you're not going to do anything, because what are you going to do? You have nothing to explain, so you just go do something else, I guess, right? So before anybody bothers to do any science about how long these things can survive, right, somebody's got to make an observation that these things, hey, we found them in stuff we think 70 million years old. How can that work? Now we have an observation. We can formulate hypothesis. We can test, and we can do everything else. But until then, why bother? Why bother testing every imaginable scenario to see if maybe it can get these things to survive that long, when you don't have any reason to think that it would anyway? It's just it's nonsense. So first, people had to find it, and then you find out how it could have happened. It's just nobody tried, because why would they?
Well, I mean, you're also dealing with somebody who doesn't know what a protein is. He doesn't know what amino acids are. He doesn't know any of these concepts whatsoever. And that's why when you ask him for, like, okay, what's the science by why these things would fall apart, as you're asking him, he doesn't know how to explain these things. I mean, he's got an entire education from listening to Neffy. That's where he started off. C. Brown was listening to Neffy in his rooms.
Yeah. You see, this is one of the reasons why I specified the topic, if I was going to have a structured debate with Neffy, that it would be on that paper. And it's because in any debate with Neffy throws out a whole bunch of references. And with most people, you would trust them, that they're telling you accurately what the reference says. But because of what Neffy did with this one, you can't. And the whole point of me specifying that debate was to actually show just how much he had misrepresented what that paper was actually saying, and then having done so means that nobody should even bother getting into debate with Neffy if you cannot trust his sources. Well, you cannot trust what he says about his sources. When somebody is at that level of, I'll call it incompetence, but I have my suspicions, what's the point?
I think it's even worse that
What
C.
is the
Brown.
point?
I think it's even worse that C. Brown thought the argument had to do with, wildlife, right? And he's like thinking that the argument really was, oh, look, there's disease risk associated with the translocation of wildlife. That to me is even dumber than Neffy did in some ways.
Yeah, he did exactly the same thing.
And
It's
I
not
think
worse.
that it's really weird to me, because he's trying to set up a debate with Neff, and I don't know why he's trying to set up a debate for Neff when Neff could just do it on his own. But that's not the point. Trying to set up a debate between somebody and Neff, when Neff evidently, one, can't accept correction on an obvious mistake when it's made to him immediately, and two, can just throw out randomly a link without bothering to check to make sure it's the right one.
I think he has
right?
acknowledged.
And then take weeks.
I think
take
he has.
weeks.
He has acknowledged, CRISPR, that it was a mistake. He had the wrong paper up there, but that's a bullshit
Yeah,
excuse.
he did.
It's a
You know-
bullshit. Yeah, it's
you
a
know who
bullshit
he-
excuse.
Hold on. No. Wait, hold on. Wait a second. Do you know who he admitted that to? He admitted that to C. Brown in an email
vaguely.
to C. Brown that C. Brown then posted elsewhere. I don't think, Fiona, did he reach out to you and apologize to you for giving you that link when it was the wrong one?
No,
I didn't think so.
What about
right?
well,
So-
Christopher? I have a video on this too. I have a long video with me talking to nephew about this very thing. He didn't. I could tell you right now, he never reached out to me and said, "Oh yeah, by the way, I was wrong."
Right.
Steve, he
That
never
is
said that to me.
unacceptable behavior.
Mhm.
That's disqualifying behavior when you do things like that. It's just,
I agree.
if he made that mistake, I mean, that's a pretty big mistake to make, but fine, people make mistakes. If he did that and it was the wrong one, then he goes to Fiona and says, "Hey, I gave you the wrong paper. It was my fault. I'm sorry." And yeah, it's it's a pretty big mistake, but I guess we get over it. But it's just
Nephew
really
hasn't
bad.
been around for
And
a
the
long
thing
time.
is, though,
anyways.
when he made the mistake, it was in a video that he was making, and he had the full abstract on the screen. That's how we all located the paper. He had the full abstract on the screen with the first line saying that translocation is defined as.
Yeah, that actually, I don't remember actually seeing that. That's just unacceptably bad. I mean,
I've got a clip of it in my video
it's
of him sharing the screen, and the whole abstract is there.
it's one of those things. Like, I try to be extremely careful whenever I'm trying to give a link to something to make sure that it's the link to the thing I remember, and I will follow that link right away as soon as it's posted to make sure that everything, the URL survived and everything.
Well, yeah, but he didn't care one way or
He
another.
wasn't
All
giving a link, though. He
he
wasn't giving a link.
he had
He
the
didn't give a
abstract.
link.
It's like I'm and if
He
I'm going to show
screenshot
it,
the abstract.
if I'm going to screen share it, I screen share the window, not my screen, because I actually know how to use Google. And I'll make sure this is okay. Wait, I just opened this in a new window. Is it the thing I want?
CRISPR, you got to remember,
check
he
it real
only read the
fast.
abstract. Not the abstract. He
No,
only read
he
the
didn't
title.
read the
Sorry,
abstract.
sorry,
He
sorry.
read the
Let
title.
He
me
screen
correct.
shared it apparently.
No, he only read the title, and he didn't even make it past the first couple words of the title. The words of the title were disease risk associated, blah, blah, blah.
See, I understand that. I understand
With
what I'm
translocation.
saying is, if I'm going to screen share something, I'm going to open it up in a new window. I'm going to go to that window. I'm going to look at the thing. I'm going to make sure that it's the thing I want and that it talks about what I think it does.
Yes.
And then I'm going to hit the little screen share button. I'm going to select that window to screen share.
Right, but he thought that paper literally was about genetics.
Yeah, because he's an idiot.
Yes.
Yep.
Because he didn't do due diligence to make sure it's the one he wants.
Exactly. That was the funniest part.
But you see, that's the thing. He's now claiming that all he did was screen share the wrong paper. But I don't think that at the time he realised that that paper was not about chromosomal translocation.
You can't do something like that.
I had asked him for 2 hours in another hangout, and I can't remember the person's hangout. I made a video about this again, asking about this particular paper and explaining to him this was about wildlife for 2 hours, and he wouldn't acknowledge it. So yes, he knew. I mean, he was being purposely evasive. At that point, I think he knew that he had fucked up. But if you watch this video, I'll see if I can post the link. You can tell that he was being evasive. Every time I asked him, "Hey, Neffy, what is it about this paper dealing with translocation of wildlife that has to do with genetics?" And you can see every time he was just being so defensive on it, but never acknowledging that the paper had to deal with wildlife translocations and not genetic translocations.
And I mean, making a mistake as big as that one, if he couldn't see what the mistake was until it was pointed out to him, and he was still denying it for quite some time, until he could deny it no longer. And also, he kept coming into Steve's hangouts and just disrupting them, challenging me to a debate. He kept doing it. And he would have a spiel to say, and you couldn't get a word in edgeways. And then he would basically say, "Oh, right, so that's an oh, yeah, I thought you would chicken out," and he would leave. And nobody get a chance to say anything. So that is why I issued that challenge. And suddenly, Neffy stopped coming in and disrupting Steve's hangouts.
Yeah, he was doing hit and run.
Yeah.
The link is
And he
in the
just
live
stopped
feed.
coming
Yeah, let me put in the inside too. Watch this video when you get a chance, Chris. If you haven't seen it, it's called Neffy Owns Himself.
Right. Yeah, can I,
Yeah.
if we have a chance, something about Neffy and young Earth creationists in general.
Nuts.
Recently, I brought up the case of Niels Herbert Nielsen again.
in. Oh, yes.
I have the book right here, Fiona. It's open to the right page. Let me explain what's going on first. This used to be one of Neff's favorite things to bring up. He goes in cycles with stuff he brings up. He'll bring up something again and again and again, then he'll drop it, and then he'll bring it up again. Well, it's his story from time to time. And he loved this quote from Nielsen, and I'm going to give it in just a minute. But he started off by saying that this is a scientist who studied bacteria for many years, and then he went on to say that Nielsen was a bacteriologist. He's not. He's a botanist. This book in 1953. Now, let me give you the quote. I'm going to give it to you from Conservapedia. Young Earth creationists use this quote all over the place. If you look Niels Herbert Nielsen and 40 years, you'll see it all over the place. Let me give you the quote that they give. And there are 3 dots at the end. There's an ellipsis at the end, but sometimes they don't do it. So here's the quote from Conservapedia. Demonstrate evolution by an experiment carried out for more than 40 years have completely failed. At least, I should hardly be accused of having started from a preconceived anti-evolutionary standpoint. You see, and there are 3 dots after that. So that's the quote you see. And if you find a decent source, they will tell you, as Conservapedia does, that it comes from his book, Synthetische Arbeitung, sorry about that, from 1953, and it's on page 1185. All right, I'm going to go to page 1185 just to find these books. And at first, I kind of wondered why he was quoting from it, because the book's in German. Turns out there is a part of it that's in English. And I'm going to start on page 1184, and I think you'll find this interesting. In this case, we have in fact succeeded in creating a new Linnaean species, which is also very definitely a positive novelty, since it contains several times as many genes as any other species of the genus Salix. Salix is a willow. Okay, that's my interjection there. Well then, is there not here a striking example of an experimentally produced evolutionary step? From a morphological and cytological and also from a genetical point of view, Salix polygena fulfills all the requirements of a step in progressive evolution. The question remains before a final decision may be made okay. The question whether the new species is vital enough to survive under natural conditions. I have often returned to this question of vitality, and obviously it is of a decisive nature. Polygena in several localities. In tilled and richly fertilized garden soil, some individuals have grown very well, attaining a height of 3 meters and apparently being quite vital. Been sown after a few years, so that some similarity to natural conditions has been obtained. These plants have rapidly lost in apparent vigor, and several of them have soon died. Individuals of the parent species growing close by have still been quite vigorous. Of the original hybrid individuals is still poorer. The plants all show such signs of degeneration that it is evident that a polygena population would never be able to survive as part of the wild growing flora. It's unable to contribute to a further evolution. My attempts to demonstrate evolution by an experiment carried out for more than 40 years has completely failed. Anyway, I should hardly be accused of having started from a preconceived anti-evolutionary standpoint. No doubt the lack of vitality in polygena, as well as in common hybrids between 2 species, is caused by the interactions between the genes of the different species, which have widely differing physiological and ecological effects. During a long time, the old species have been the subject of natural selection. They have been bred to harmony with the environment. combination of their genes is therefore an intergenic catastrophe which must result in the avitality of the progeny.
I have no idea what you just read, but that's okay.
Okay, let me
I do.
let me try to put it
Yeah,
succinctly.
what's the
Okay. So Dr. Nielsen created a new species of willow
uh-huh.
that he named Salix polygena. Okay. And he created this new species by, it explains it up here how he created it, by crossing. Okay. And he looks at it and he goes, okay, looking at it, usually if you look at the genes, if you look at the morphology, if you look at the cell structure, that's the morphological, cytological, and from genetical point of view. Okay, so if you look at it all three ways, you got a new species. But then here's the next question. Even though it's a new species lab, what about out in the wild? Does it retain its vitality in the wild? So he took it out in the wild and he tried to plant it. He's a scientist and he has the results here and he explains how he planted in different environments and things like this. Survived. It did not have vitality. So because it did not survive, he said, and also he said that my attempts to demonstrate evolution by an experiment carried on for more than 40 years has completely failed. He's talking about this one specific attempt to create an artificial species. That's what he's talking about.
That would have been the setup initially before you read all that because I didn't know what you were reading.
Okay. Well, I think Fiona got it. Some people would get it, but that's okay.
Yeah, I wasn't.
And then he goes on. He said, "No doubt the lack of vitality in polygena is caused by the disharmonic interaction between the genes of the different species." So, in other words, what he's saying is, he took these different species and from them created a new species, but the genes don't mix properly to make a vital species. This statement has to do with this one experiment. It has nothing to do with evolution in general.
They took them well out of context and twisted it to their own narrative.
And by the way, I'm going to I haven't done it yet, but I'm going to take pictures of these two pages because it's actually on two pages, 1184 and 1185. I'm going to take pictures of these two pages and post them and use them for reference if they ever see this anywhere because it is brought up. But this is wildly text. And again, this is only 2 pages. He explains the experiment and so on and so forth. And then at the end he says, well, my attempt to create an artificial species failed. Has nothing to do with evolution in general. And it sure as hell has nothing to do with bacteria, as Neffy thinks it does.
Yeah, it's amazing their ability to twist this shit, isn't it? I mean, just mind-boggling. Yeah,
and Nielsen, by the way, the one thing I will say that let me read this from Conservapedia because Conservapedia occasionally gets stuff right. Oh, and by the way, when their references, they reference another site that references the original book, which is not the way you do things. And this is Conservapedia is notorious for this. I've been through them on their and also on evolution. They don't quote. This is also, by the way, common with evangelicals. Matt Slick does it. I've seen other people do it. They don't quote original sources. They quote somebody else who then may quote the original sources. Answers in Genesis does it. A lot of the what I call the professional creationists do it. Answers in Genesis, Creation Mysteries International. They don't quote original sources. They'll quote somebody else. Original sources. That's just not the proper way to do things. But anyway, they say Dr. Niels Herbert Nielsen was not a creation scientist, but was a founder of an evolutionary hypothesis called emication, which is described by review in the Quarterly Review of Biology as stating that at various periods in geological time, Violent revolutions have destroyed all of the Earth's biota, only to have living forms reconstituted by a sudden coming together of organic molecules to form gametes possessing the capability of developing into some highly complex form, such as a pine tree, an elephant, or a man. Yes, an excellent scientist. I found references showing that he was an excellent teacher. He was a little eccentric and had his own unique version of evolution, which is why he wrote this book, because this book basically means what? Synthetic something or other. I forget the translation, but yeah, it's about his own unique version. There's I don't really even know if there's any evidence for it because I haven't bothered to study it. But yeah, so they take things wildly out of context. And they don't care, and nobody ever seems to bother to look these things up.
Well, this was before they even discovered genes, wasn't it?
He published this the same year that the structure of DNA was described.
Yeah, I mean, so.
Oh no, they knew about genes.
They knew about
They
okay, they
didn't
didn't know the
know a
DNA
lot
structure.
about it.
Yeah.
But you see,
Right.
this is it just highlights yet again how cavalier Nephi is with his sources.
Mhm.
Now, in a structured debate, you can't go away and check out all of his sources. And we have two examples of him dishonestly quote mining.
Mhm.
Or he is a true example of citation bluffing. And in an oral, live, structured debate, you don't get the chance to go and check out all the sources.
Yeah, and let me point out.
But in a written debate, you
Right.
do.
And I wanted to mention what I said at the very beginning. This book is actually in 2 volumes. Me a couple of years. I'm not really sure how long it took, but I'll just say a couple of years at the minimum to find these books. To me, from Germany. These books, if you look at WorldCat, for those of you who don't know, is a place that shows you where books are in libraries all around the world. You won't find these books in very many places. I would have had to have gone to Stanford or the University of California, Berkeley, to see these books. And this, by the way, is another problem I've discovered with some young Earth creationists, is that they will quote obscure sources that it's hard to get a hold of. And that is why, again, like what Fiona said, they throw this stuff out. You have to be able to look it up. And in some cases, like this one, it's an obscure source that it is difficult to get a hold of. I can't go online. There is nowhere. Well, actually, there is one place online where this book is copied, but I have to digitize. Thank you, digitize. But you have to belong to this group to find it. However, you can search. But when I tried to search, I came up with nothing. Didn't help. When you use obscure sources, when you just throw stuff out there, people have to be able to look it up.
Mm-hmm.
So I have these books. If anybody ever brings it up, like I said, there's another quote from another page, and I'm going to take the pictures. I mean, I'm sure 1953 is probably out of copyright or whatever. I don't care. For 2 pages, I should be able to have the right to do that anyway. So I'm going to take photographs of these and put them online. But that's why I won't do a verbal oral debate, because they throw this stuff out. Not only that, they throw it out. They gish gallop, throw out one thing after another. And for a while there, a year and a half ago or so, I would listen to some of Nephi's hangouts, and I would stop them every time. I mean, he would spew out a list of stuff. Right, okay. I would stop every time he spewed something out, and I'd write it down. I had a Word file open. I'd go and type in the Word file. Quotes that he gave out. He would give a quote. He would say, so-and-so said this, and I would type out the quote. I spent some time looking up. It's almost all wrong in one way or another, in the same way that this one was. He's misquoting people. Wrong. He's referencing. I actually got him to admit to me once in a hangout that he doesn't do anything other than read the abstracts. He said because he two things. He can't afford to look them up. Although I've pointed out to a lot of
I
people,
just-
there are things called libraries, and
I-
you can go there.
I just need to interrupt you very quickly. I don't believe he reads the abstracts.
But his excuse was everything you need to know is in the abstract.
Well, that's just stupid.
I know.
Yeah, I mean, that's a good
Well,
summation.
and especially since some abstracts can be misleading. You know, an abstract might say something like, here we look for X, Y, and Z and find out if A, B, and C are true. And then when you get to the bottom, it says, hey, guess what? We found A, B, and C aren't true. We'll read it, go, look, A, B, C, and C are true. See, it says right here in the abstract, they're true.
I mean, if you don't even read the conclusion of the paper, then I mean,
No, he doesn't.
and see, I guess C. Brown is throwing out more accusations now. I guess now he's telling CRISPR that iron can't preserve in deep space time. I think it just means deep time. Why are you calling it deep space time? It's just deep time. But he asserts that these things cannot last. And then what else he says in here? Oh, you can't change science. I love what you put here, CRISPR. There was no science on the specific conditions in question, so nothing needed to be changed. Bingo!
Yeah, he doesn't.
That's brilliant. That's awesome. That's exactly it.
He still does not. Well, first, he doesn't understand what my original point there, which was that he gave us a link to a creationist, giving us a link to what I swear was an undergraduate writing a paper. That's unfair. No, I mean, I'm sure it was a graduate student. Doing a very, 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 very shallow overview of preservation of biological molecules, in which they referenced somebody else's unpublished data about a completely different condition that has no relevance at all to the specifics here. And he doesn't get it. Like, you can't.
He doesn't
just...
understand that these are specific conditions that allowed for this kind of preservation. And if he wants to try to falsify it, he needs to come up with an explanation to show that under these given conditions, it would not be possible.
Yeah, it's just I don't even know.
Well, that's what I'm saying. I think he's absolutely dishonest at this point. I mean, it's just blatant that he's sitting there saying all these accusations and assertions, and yet nowhere in this thread has he provided a single link to anything that actually substantiates what he's claimed. Not
You know what this kind of reminds me of? It kind of reminds me of the way people like to say the thing about bees not being able to fly, right? Because at one point there was somebody who said, well, based on this very very narrow model, right, it can't work. But there was nobody at that time who did an expanded model that would cover everything, right? So that's the way this works. You don't have everything all at once. You expand and you add to explain ever more information, and eventually you cover it, but it wasn't covered then. So what are you going to do?
Did figure out how bumblebees fly. And just as a little interesting aside, my dad was an aeronautical engineer, and he designed. He started working on airplanes, and he worked on airplanes during the war, which is World War II we're talking about, which is why he didn't fight in the war. He was too busy designing, airplanes. And he told me at the time that, we talked. He mentioned the thing about how people couldn't figure out how bumblebees fly, and it was actually only ago that they completely figured it out. And he said the engineers were really puzzled by it, so a group of them decided to build aircraft, a model aircraft, not like a full-sized one, built along the lines of a bumblebee, and much to their surprise, it flew. Figure out how it flew, but it flew.
So, just to go back to the fact that Neffy apparently is fond of quoting obscure articles that are hard to get hold of, isn't he self-refuting right there? I mean, if he's trying to make claims that science believes something ridiculous, surely citing really obscure articles from long ago is just no way that there's no way that could work. And what's your argument? Hey, there's an incredibly obscure article from far in the past, and therefore modern science is wrong?
Well, sometimes they do, and sometimes they get that wrong. Because I like to joke I spent a fair amount of time in the 19th century. Thanks to Google Books, I spend a lot of time looking up stuff. I'm interested in how people looked at science in the 19th century. The word scientist didn't exist century, and many people, including Darwin's bulldog, Huxley, hated the term scientist. He actually equated it with the term cannibal, I believe. So, the words used were different back then. And for some reason, they try to say, well, this wasn't always so. They actually contradict themselves. And on the one hand, they'll say, well, this isn't the way this was always done. An example is, this happened last time I mentioned this, Steve. Vestigial organs. They will go and say, well, you see, scientists have changed the term to vestigial organs to fit their new idea, so they can say that it still works. It's as if never changes, never grows. So that's one thing they'll say. They'll say, well, see, it was always this way, and then scientists suddenly in recently have changed it just so that they can't be wrong.
Well, yeah, that's the whole point, though, isn't it? You're trying to improve your understanding and your definition, so you are no longer wrong.
Right. Except that when you go back and you look at the actual literature on vestigial organs, which I did, you will find that there was quite a bit of discussion over what is a vestigial organ and or a vestigial structure. Organ is really the wrong term, although that's a term you often hear used. What is a vestigial structure, and how do you define it, and should these things be included, should these not? So it's no, I'm sorry, there wasn't some 19th century exact definition of vestigial structure that scientists have changed. So, but on the one hand, they try to say the past is correct, but on the other hand, they'll go to the past for something obscure and say, well, you see, they were wrong. Scientists have been wrong all along. So either the past was correct or the past was wrong. Stupid argument either way, because as you point out, as everybody knows, science changes and it evolves. But none of these people bothered to look into the past. None of them bothered to look at the journals. And you can go to Science and Nature, and they have their editions from the 1800s online. You can read them. I've done it. I've read the original reports on, for example, the Nebraska Man, and it's easy to do, but they don't bother to do the research. They just go with what somebody told them that they got from a website that borrowed from another website that is referencing still another website that's referencing some source that we.
One of the things I really enjoy is lectures about the way in which the scientific method, the method has evolved over time too. It looks like, for instance, there's almost a paradigm shift in how we treat the science of how humans interact going on at the moment. Whereas I guess in the hard sciences, we were lucky enough to have simpler problems to solve.
One of the fun things to do, though, too, is to look at how things have changed. I've spent some time, and I'm going to do a video on it in the future. What reptile means, because we know that the concept of reptiles really, really changed recently, and I've heard that argument too. Is, well, what, you don't even know what a reptile is anymore. Well, if you go back and you look at the early literature, you will find frogs listed as reptiles. You will also find crocodiles listed as lizards. They, depending on who you were looking at, you're looking at early works and people trying to figure stuff out. That's basically what it is. Trying to figure stuff out. And because crocodiles and lizards kind of look alike, they were put together. I'm not sure why frogs were put in with but they were. And it just, you can find these things, and it doesn't mean that they were wrong, because if you bother to read what it says, you will see what criteria they have for putting this thing there. The one with lizards and crocodilians is kind of obvious. Look at what a lizard look like, what a crocodile looks like. Even the way their legs are structured is similar. So their heads is similar. And of course, other people would describe them differently, say, no, you have to put them in two different groups because of this, that, and the other thing. Process.
So, are lizards and crocodiles from a fairly recent common ancestor, or is it convergent evolution going on there?
It's convergent evolution, because crocodiles are more closely related to birds than they are to lizards. And we've only discovered that recently. But there's lots of convergent evolution.
Yeah, and I guess I was just change the subject briefly. I was just thinking about someone said that Nephilim Free was talking smack about Ms. Schweitzer, and I was just boggled that anyone would disrespect someone who had basically ushered in a maybe paradigm shift is too strong a word, but she changed the game certainly.
Well, their argument is that, well, they don't understand what she did. So they think that she found real soft tissue that was soft, not understanding to begin with that soft tissue was a type of tissue. And it doesn't mean that it was soft. They had to demineralize it. They had to put it through these baths to get it. They don't understand what she found in the first place. And they think that she found real red blood cells and soft. I actually at one time tracked, and I did not save the links. I should have. It's one of those things I regret. I tracked backwards something that I don't remember, it was Neff or someone else said. Information from a creationist site, which got their information from a creationist site, which got their information from a creationist site, which got their... Okay, when I tracked this backwards, I discovered this is turned out to be like the what do you call it, like the telephone game, or where one person whispers something to another and then they whisper to someone else, and it changes along the way, right? We used to do that when I was a kid. The nuns would have us do that to teach us how gossip works and why you shouldn't do it, because the time it goes around the circle of kids and comes out, it's a completely different story.
Yep, played the game.
You've played it?
Scout camps and such like, definitely.
Yeah, and you see the first statement comes out completely different because people mishear. Well, I found out that's what happened. One of the things I discovered, because the very first reference was good but slanted. They had the actual information, but they put it in a slanted way. As it went from step to step, it more and more. For one thing, one of the most interesting things is in the first reference, they showed pictures from one of her papers, and it had this blood red stuff. Also showed the picture that showed where she is comparing the material she found with the ostrich bone, right? Well, it got to one point where these people apparently didn't pay attention to what was going on, didn't read it, to the ostrich bone as being the dinosaur bone.
That's not a bit surprising.
Hey, let me ask you. I'm
So-
going to ask Jade a question. Jade, in her papers, she does have a lot of the blood vessels from ostriches, right?
Right.
And there's one picture on ICR that I don't know if it's. There's actually two pictures on their page. One is the top one, they say it's an ostrich. The other one say from a T-Rex, and I don't know if the second one is from a T-Rex or not. I don't know. Now it matters to me not, because the thing they're claiming is a red blood cell in there. She's already said they don't know what these structures are, but I want to know if that image, because I tried finding that specific image in her paper, I couldn't find it. So
Give me a link to the ICR because I have her papers. I think I know which one you're talking about, and I think I can find it.
Yeah, I'll do it right now. One sec.
Okay. And one of the other things that some of these people don't know, because I found one slide in which they're going, see, it's red. And I'm laughing my head off because it's called a stain, folks. Do they understand that when you're looking at a microscope, you stain things using different stains to bring out different features? So there is one in the creation universe where they are claiming that it's red blood and it's a stain.
Well, I remember
down to its
there's
original.
iron, but which one's a stain?
Well, no, this isn't that one. I don't.
Okay.
This isn't that paper. I don't remember which one this is. And again, see, the problem is, I wish I'd have kept the links to my little, because the thing is, I was going from one to one to one very quickly, and I didn't keep all the links of my exploration backwards. And I wish I had because it would have been a great, video to show how they'd gone it up along the line. But I don't know if this one's from Mary Schweitzer, but I did find a picture where they're going, look, red, it's a dinosaur. No, it's a stain. It's a stained section of dinosaur bone. And I don't even think it's Mary Schweitzer's. I think it's something else altogether.
I put in the inside chat for you. Can you look at that real quick?
Okay, it's just I will.
Second image. Yeah, well, I mean, this is something that's common along the ICR and the IG thread is that they're just so dishonest to people. And again, I know before I was like trying to give people the benefit of the doubt. I'm not doing that anymore this year. I think these people are outright lying. I don't trust them as far as I can throw them because, Dr. Tompkins especially, before I was like, I don't think he's lying. Maybe he is. I don't know. Now it's like after I've talked to him and corresponded with him, yeah, I don't think he's very honest because I've looked at the things that Ruhif sent him and the corresponding letters back and forth with them, and I mean the guy has to know that he's not able to substantiate what he's saying. He has to know this. I mean to have that level of qualification and be that completely ignorant, I don't know. If it's not dishonesty, it's the worst case of cognitive dissonance I've ever seen in my life. And he's the one that's saying that Ruhif doesn't have any qualifications, yet Ruhif is the one that explained to him his errors. Come on.
Really unfortunate because I was really looking forward to being in a room with him.
I know. I tried. Well, he said he was going to, and then I said the person at ICR struck that down, and then he actually did you listen to the email that he sent back after I said this is unfortunate because we kind of want to hear it from your own words, but let me see if I can find you his email real quick, CRISPR.
I think I saw it.
Well, it's just the tone had changed completely. He said, FYI, oh, by the way, I said, I will be having a video about this in a week or so, and we'll link it to Dr. Tompkins for him to provide a rebuttal if he would like to, as I believe that is only fair, right? Nothing, hostile about that. And he said, FYI, I have a peer-reviewed paper coming out in about a month that is a response to claims by Ruhif and others. I would suggest dropping this whole effort until that paper comes out. Also, Glenn is not a geneticist or a biologist, and many of his claims he has made were easily debunked. Well, there's a few problems with that. One,
up until he said easily debunked, I don't necessarily have a problem with anything there.
Well, the first part,
I don't think.
Well, I do have a problem with the peer-reviewed paper because he's not. I'm pretty sure he means.
We know what they mean.
Okay, and the second thing is he didn't easily.
His point about waiting until.
I'm sorry. What?
Go ahead. His point about waiting until his new piece of work comes out, I think it's fine. I mean,
I have no problem with that.
Sure. Yeah, and then it's true. Ruhif is not in the biological field, right? That's fine. So, okay. His claims are easily debunked. Well, no, they're not. He's right.
Well, here's the thing, and when I responded to him about that, I said, we do have many PhDs in biology and bioinformatics in our community who are geneticists and biologists and who examined Glenn's data and validated it to be sound and asking the exact same questions about your work as he has pointed out to you prior. So, there are people in this community that do know about these topics. Even such as somebody like yourself, you've looked at Ruhif's data. Do you find it to be subpar and erroneous and easily refutable?
I've not seen anything about Ruhif's explanation of this that's wrong. It's honestly, I did a lot of it on my own and came up with the same results. So that's take that for what you will. Obviously, I'll take a look at the paper when it comes out. The blog post, I mean.
Yeah, I'm looking forward to it too. But then again, after it comes out, I'm just going to be messaging him again. Okay, we've read this. We're going to have a hangout. I'm sure. I'm sure it'll be brought up and talked about. And then we're going to have questions about that probably. So it's going to be an ongoing thing. At some point he's going to have to like step in and say, okay, I'll go on air and do it. But
Try to be nice to him though, because I mean, he's
I am
not going.
nice to
He's
him.
not. Yeah, just I mean, let's keep the tone. Let's keep the tone friendly. I don't want to say that he's dishonest. I think he's wrong about things, obviously. But I'd like to have that conversation with him and just to see what he has to say about some of the criticisms that I have and Ruhif has and other people have. And just to see what that reaction is, because
I would
I'm
love
pretty sure I
that.
know what it's going to be, but you never know.
Well, I mean, there was two questions I already asked him, and Glenn actually had fielded them already, and he said, I know how Thompson's going to answer, but Thompson is wrong in his answers. He uses these other types of databases to try to imply things that just aren't there. And again, you think he's not being dishonest, but for somebody to have a PhD and be somebody who does know what he's doing, I mean, everybody said he's good at his research. I don't know how anybody can be this
Well,
deceptive
I mean,
and not be
what
willful.
I said was I don't have any evidence that he is being dishonest. He may have just made a series of very significant and really just terrible mistakes that would be hard for anybody with that level of training to have made. But right, so I don't want to say that he's being dishonest because I cannot demonstrate it.
I know, but it's a gut feeling.
I mean, it's one of those things like when he's publishing something talking about the chimpanzee genome, and he's not using the most recent assembly. He's using an assembly from like 4 years ago. Why would you use the assembly? Why would you use an out-of-date assembly, right? It's probably because it's the one that's got that intentionally inserted gap, isn't it? Oh yeah, that's probably why.
The 140 some odd thousand base pair thing.
Yeah, because it best suits his idea or presupposition or whatever. I mean, you notice a lot of them will go back to old data. It could be 30, 40 years old. You go, well, see here's what it says. See, y'all wrong.
But that gap was artificially put in there to make it help, look like to see the alignment.
It's not a gap.
Well, it's missing. It's
not a gap. It's not a gap. It just isn't.
How about missing base pair?
It's just no. There's no missing base pairs there.
Removed base pair. How do you want to phrase it?
It's an alignment issue.
Yes.
Gap, missing base pair, all of that, removed base pair. It implies that there's something there that we don't have the data for.
But we have the data for it. They just didn't.
Right. Yeah. Because there's nothing. There is no DNA there. Okay. In that early region that goes on for hundreds of megabase. Right. There's no DNA.
I get that. I totally understand it's an alignment issue.
Because it hasn't started yet.
But he knows that too.
Right. So when he says right. So if he calls that a gap, he's lying.
Okay, I got you. I'm on board. I wouldn't be a gap because we do have that information, but he has a PhD in this. So don't you think he's a little bit duplicitous if he tries to imply there's such a huge amount, quote unquote, missing, which not missing because, like you said, we know what it is.
It's pretty bad. It's pretty bad when you consider the fact that he's saying that we have. Just quickly doing the math in my head here, 300 and something megabase from the chimpanzee 1 and 2, compared to 240 from human chromosome 2, even though there's only about 240 megabase in chimpanzee chromosomes 2A and 2B. Well, wait a minute. Something weird just happened there. How did you just go from 300 and 300 and something to 240 something? Oh yeah, it's because you're not using the numbers right. Like, it's a fairly severe kind of incompetence.
Well, what I am in touch with, I have a few people that I've reached out to that are other creationists that may be able to defend Tompkins' work. Again, it's not the same thing, but at least it's entertainment. So maybe we'll find somebody kind of like Ben tried to do, right? I mean, but he failed miserably. But I think it's entertaining. I think people do enjoy that. So I have a few things in the work of people that I've been talking to that said, yeah, I know I'm familiar with Tompkins' work. I can explain it. Okay, well, come on in and give it a shot. Again, not as good as Tompkins himself, but it's still fun because I like to listen to these people try to explain this shit when I know full well that they can't, and you know full wrong right, that they can't. So, anyways, it's about time I bring this to a close. I'm going to go play a little bit on my mage, get some PvP gear, and maybe Joel's G will join me at some point. PvP is my thing, but see the problem is Joel's is that I'm so even though my gear doesn't matter. I'm undergeared in the sense of honor talents, right? I'm only at like tier 3, obviously, and so there's like 50 tiers. So it makes a huge difference how fast I get my ass kicked. So no matter how good I am at PvP, it makes no difference because I'm 6 weeks behind the season. So I've got to catch up.
I feel like all of the air in the room was just replaced with nerd.
Yeah, that was nerdy. It's the ending of my hangout. I'm going to nerd out. I think those PvP honor talents get reset every season, don't they, Jules? But I think you just got to keep on building them up. But you should go play it, CRISPR. Try it out. It's good stress relief. I'm just like, okay, yeah, kill C Brown. Just picture all them all as C Brown. So, but anyways, anybody last words before I take this off air? Oh wait, did Jade leave? Is she still here?
No, I'm.
Did you look into that?
I'm still here. Yeah, I'm gonna have to send you the link when I get it because my connection is not working really well and it keeps going to.
Yeah, my computer's not working very well. My computer's been lagging, dropping frames. I mean, it's just dying on me.
Yeah, it's the weather here tends to really affect things because our towers are on top of the hill here and on top of the mountains, and it just really screws them up. Being in a rural area too, I don't have a lot of choices. So anyway, I found one link to her a Smithsonian article that I had lost, which is kind of cool. So I'm going to read that first, and then I'm gonna. It has the same picture. That is a picture. Given that these not Smithsonian. Is it Smithsonian or is it Smithsonian? I think it is. Yeah, Smithsonian. Given that the Smithsonian is using it. Yes, that is indeed a picture of the T-Rex. It's the same exact picture that ICR is using. And the question I have is, how did they get that picture?
It says used by use as free, fair use.
Right, I know, but what?
I don't know where they got that picture from.
Well, what conditions? I will find. I think I know which paper it's from. But I'm going to read the article from Smithsonian.
I looked at 2 of her papers, and it didn't have that exact picture in either of them. So maybe I
know which one it's from, and I think I have it, but I'll find it. I have public links to most.
Yeah, like I said, I probably would normally accept that it was the T-Rex. I have no issue with that. But it's just when it comes to some sites like ICR, I don't trust them at all. So.
Right, and especially if you will notice, they say under this is again typical of creationist sites. Will say that the picture is copyright 2005, right? And yet in their references at the bottom of the page, they don't reference anything from 2005.
I didn't see a reference for it. That's why I couldn't find where it was from. They didn't give a reference
exactly.
For it. Right.
No, and they don't bother doing things.
And I noticed that. That's why I was questioning it because I'm like, look, there's no information about this picture that I can find. And again, I'm not questioning if it is or isn't as far as being a T-Rex. I just want to know where it came from.
And this is a problem again with discussing things with younger creationists. If they give you a reference like this, my response would be, whether it's a T-Rex or not, I reject your reference. Your reference doesn't have an actual reference.
Yeah,
That
it's not
paper
properly
doesn't have a
annotated.
reference,
Right.
so I can dismiss it out of hand because there's no reference.
And that's a brilliant point too. I mean, that's what I find consistently with these ICR articles.
Yeah, and I do too. Yeah.
And when they do annotize something, it's usually self-referential
Steve,
and vague at best. Yes.
ICR blog posts.
Yes.
Let's be careful.
Yeah.
ICR
blog post. Yes. They cite blog post.
I
No,
don't remember.
those articles are not articles. They are blog posts.
They're blog posts.
Yeah.
You're right. They're
Right.
not really
Right.
articles.
Okay. Yeah. Well.
I don't remember. Just briefly tell you, there was one thing I don't remember. If it was ICR1, but they had this thing near Halloween a couple years ago called 13 Grims for Evolution. Like 13 things that knock down evolution. To begin with, it isn't even 13 because they're repetitive. So in one case, just got to tell you this before we go because it's funny. So they give you a reference, right? And the reference is another one of their articles. Okay, fine. That article gave a reference to another article, right? So let's say I'm starting with the Grims is we're going to say that's article A, okay? Reference to article B. Blog post, excuse me, you're right. So blog post A referenced blog post B. Blog post B referenced blog post C. Blog post C referenced blog post D. Blog post D referenced blog post. You got that?
Yeah.
It's circular.
Yeah, I get that. But if you
and it's like,
but
well, where did it come from originally, you dumb fucks? You're just going around in circles. BCD, BC, BCD, BCB, BCD. Jesus.
Well, that's why that's why I question the citation here because again, if you look at the fourth references, as you said, none of them have anything to do with 2005.
No.
The oldest one I think goes back to 2011. So yeah, I don't know.
Right. So my point of view, if someone were to give me that, I would dismiss it out of hand. It's not real, but I can dismiss it out of hand because you've given me no reference to check on. That is a section of bone. Bone, for all I know, wait, let me see. That section of bone, for all I know, could have been stained, and that's why it's that color. No reference to go by.
But see, they're saying it is a red blood cell.
I know, but I can't. I have no reference for that.
I don't either. I can't. That's why I want you to check it in for me because I
know, and I can dismiss it out of hand. I can even dismiss the color out of hand because I don't know if that's a stained section or not.
Right. Of course, we can dismiss it out of hand.
Yeah.
But can you do a little more research on it? Because again,
I
will making a false claim here. They're saying that this is a red blood cell in a T-Rex, and that's not in any paper I've ever seen of hers claiming that.
No,
so the photograph might be legit, but that's not what the claim is.
I'm sure I've seen the photograph before, and I think I know which paper it's from, and I will find it.
All right, let me know, please.
Okay, I will. I'll send you a link. It's just that my system is just, and there it goes again with the weirdness. And what it's doing is, the thing is, if it weren't for the fact that I actually have RAM and things are loaded into the RAM for over a period of time, I'd be cutting in and out like mad because it's out again, but you can't tell. But it just what it does is it makes it impossible for me to look something else up. The only thing that works is me being in the Hangout. So yeah, I'll look it up and I'll send it to you.
All right. Well, I'm going to be in the Hangout anyways.
Yeah. Okay.
Anyways, guys, thanks a lot. I'm just going to leave you guys with this. Every time you see C. Brown make a mention somewhere, anytime you see him in a Hangout, I want you guys to all ask him for evidence for his claims that Mary Schweitzer has found red blood cells and that they were definitively red blood cells. And the third one is, I'll add for CRISPR's sake, if they were red blood cells, so what? How does that affect a damn thing? It'd be fascinating, but it certainly doesn't change anything we understand about evolution or the age of the Earth. It makes no impact on it whatsoever. So I want people to bear this in mind, that these are something that I'm not going to let go with C. Brown because, again, he just throws these accusations out there that she's lying. He just throws these assertions out there that he can't substantiate. And it's about time we do keep on these people, the Clown Car Posse, with this kind of stuff. And by the way, I do appreciate the kind words that people said when they watched Ronnie's video about me the other day, and then basically was like, Ronnie was just god awfully stupid. The fact that what he put in there didn't even make much sense as far as him trying to disprove DNA, excuse me, just for him to say that DNA degrades in water, for him to say that I have said that evolution is a fact, and
I just can't believe we're still talking about DNA and water.
it's never going to go away. It's never going to go away.
That is the thing that got me into this community. Just
I remember. Yeah,
just
that
for
was
the
a
record,
talking.
yeah.
It was a Talking Shit Tuesday episode, and you had dropped in to find. You had a paper on the different bonds and what spontaneous breaking they were. You know how long it took to spontaneously break. I remember that.
Just wanted to ask if anybody was interested in that.
Yeah.
I was like, I'm interested.
It was great. It was a great paper.
Yeah.
Yeah. But anyways, I'm gonna end it. Sarah, you're welcome. Go have sex with your wife. And with that, I'm gonna bid you guys adieu.